Sanger sequencing is one method that can be used to determine the order of nucleotides in a DNA molecule. In Sanger sequencing, the DNA of interest is amplified in a process similar to polymerase chain reaction, PCR. Like PCR, Sanger sequencing reactions include DNA polymerase, DNA primers, nucleotides, and the target DNA. The primers bind to the target DNA, and DNA polymerase creates a new complementary DNA strand using the nucleotides. Then, the process repeats. Sanger sequencing reactions also include fluorescently labeled dideoxynucleotides, ddNTPs. Each of the four ddNTPs, adenine, cytosine, guanine, and thymine, are labeled with a different color. Relative to the large number of regular nucleotides in the reaction, just a small number of ddNTPs are included. As the ddNTPs are randomly incorporated into the growing DNA fragments, they halt the reaction, producing short DNA fragments of varied lengths, each with a fluorescent nucleotide at the end. Capillary gel electrophoresis is then used to separate the DNA fragments by size with single nucleotide precision. As the fragments move through the gel, a laser activates the fluorescently labeled ddNTPs, and a detector measures the fluorescence as they pass by. This process produces a graph called a chromatogram, which can be analyzed to determine the DNA sequence.